thank you very much. First of all, yeah, I really want to thank Mohammed for inviting me to this nice symposium. So I will just go directly to the outline of my talk. I will have a short introduction to the clinics, which is the company for which I work as a scientific director, and then we will move to how Zebrafish can help in all the drug discovery process, and so we will have basically three main modules. One about disease modeling through CRISPR-Cas9, and then target validation, and how then we use all these disease models for drug repurposing. Just to give you an overview, the clinics is a CRO, which is based in Barcelona. We are over 40 employees, was founded more than 10 years ago as a contract research organization. I mean, we have two main areas of research and development. There is a research and development that we do to optimize like genome editing tools or toxicological screens. I mean, we have a broad range of actually experimental settings. And then on the other side, we work with clients and collaborators which come from academia or are a pharma or biotech company. Importantly, I want to actually say that we are involved in pushing the Zebrafish system to be accepted by regulatory entities. Actually, we are involved in different interlaboratory studies with NIH and the OECD, and we have been trying this hardly because one of the main problems of the Zebrafish model, and this is a question that we often get asked, it's how translatable it's the model. How can you compare the result that you get from the Zebrafish to human settings, right? And but well, we have this double phase, let's say, in a sense that we work and collaborate with academia, but we also have clients. This is reason why we have completed well over 300 studies, but we also publish relevant scientific articles. And one of our ideas is that we are trying to build an ecosystem which is based on the use of the Zebrafish model at different stages of drug discovery pipeline. That is why we have a spin out, which is mainly based on cardiovascular disease, and it is a small pharma which was founded a few years ago. Well, our mission is to accelerate research. We, as you explained this morning, I mean, we really think that Zebrafish is a wonderful system that can really speed up the whole research process, and but is also using the Zebrafish larvae, we can have like kind of ethical way of doing research, because Zebrafish, well, as some of you maybe don't know, but they are not considered as animal models up to 5 days post-fertilization, so we can use Zebrafish larvae to work, in an ethical manner. And our idea is really to place the Zebrafish larvae, especially as a fundamental tool for drug discovery and genetic target discovery. And so let's go to drug discovery pipeline. Where do we place the Zebrafish here? I mean, the drug discovery pipeline, it's quite long and complex. Let's say it goes from target identification to the advanced clinical phase. And then, usually the target identification, it's in silico, but then there is a very high attrition rate when we go and we move closer to the clinical phases. And we really think that, the model that was used up to some years ago, it was going from in vitro to rodents, but actually most of the rodent assays then, they fail, in very early phases. So we really think that the Zebrafish can help us to narrow down the number of therapeutic candidates before going to other relevant preclinical models, which could be the rodent. And this can really save money in the whole drug discovery pipeline. Well, we are not the only ones who think that, because we have to date more than 100 small molecules identified to Zebrafish screening, and 9 are currently under clinical trials. This number might be outdated because it is a review from 2019, but in general, there are many compounds that have been screened through in zebrafish larvae, which are in different phases of clinical development, drug development. And why zebrafish is a good model to do drug screening? As you were mentioning actually this morning, the high throughputness of the model, it's really probably the most relevant feature. It has been already said that zebrafish larvae, they are transparent, they are genetically homologous to humans, and we can really perform screening them. But one of the important things is that we can use 96 well plates to do waterborne exposure of the compounds up to 5 days post-fertilization. So you can imagine that we could screen hundreds of compounds in relatively fast manner, and we can couple, let's say, this in vitro throughput, because we can treat them as if they were cells, but we have in vivo readouts. And in vivo readouts, we have many of different readouts. This is just an example of double transgenic line, which has vessels and the heart, which are labeled by fluorophores, but we can have like morphometry analysis, behavioral analysis, transcriptomics, and actually, this is what I'm going to show you. So how we couple the high throughput screening to high throughput phenotyping. And this to have just some examples. For example, we have in-house vertebrate automated system machine, which works we do have a glass capillary, which is aspiring each individual larvae, and then it's placing it under a microscope, so we can have either a bright field image or a fluorescent image. And then from these images that we get through actually deep learning algorithm, we can extrapolate a lot of different phenotypes. And what these phenotypes tell us, if indeed a compound or a genetic modification leads to some kind of defect. So let's say it's teratogenic, so teratogenic. And we can have like qualitative readouts as York edema, body axis defects, but also quantitative readouts. And we can do this, in a completely high throughput manner. And important, the interesting thing is that through this system, we can place then the larvae in another well, and then we can genotype. So we can do genotype-phenotype correlation. This is just one example when we can analyze morphometry of the fish. But as I said, we can also analyze other tissues. For example, if we look at transgenic line, which is a heart that is labeled with GFP, we can have a short video acquisition of the heart. And starting from this short video, we can have an automatic tracking of the chamber activities over time. And so we can say, if a compound induces bradycardia or tachycardia or some other different features, we can actually look at a lot of different readouts like heartbeat frequency, arrhythmias, heart chamber diameter, cardiac arrest. So we can say if a genetic modification of the compound is affecting the cardiovascular system. And if as another high throughput platform, we can look at fish behavior. Now, fish behavior, we use as proxy locomotor activity. We do assume if locomotor activity in 5 days post-fertilization larvae is impacted, then this means that there has been some problem in brain development or nervous system development or visual pathway. And how we do that also in this case, we use high throughput screening platform. Here we have visual system that you have shown actually, where we have individual larvae which are tracked over time, and then we can look at kind of stereotypical pattern of zebrafish behavior, which they stay still. Well, they move in the dark while they stay still during the light, and then we can see if there are alteration of this behavior, and then we can relate the genetic modification of the compound screening to this kind of behavior. But so let's actually, now that we talked about all the possible phenotyping platform and the high throughput screening that we can do, how we generate disease models and how we use this disease model for drug discovery. So the disease modeling, it is the process through which we have a known gene, and we know that this gene might be involved in some kind of indication of a pathology, and then we want to model this in zebrafish, for example. Let's say, of course, we are talking about that, and these. Well, as easy as that, we use the CRISPR-Cas9 mediating knockout. So we have a human patient. In this case, it's a very easy case. It's oculocutaneous albinism. So we have a patient. We take the same mutation. In this case, it's tyrosinase gene. Then we target this gene with the CRISPR-Cas9 system. So we target the ortholog gene in zebrafish of the human ortholog gene, and then we would obtain albino fish. We can have this kind of fish, but we could have, and we have generated, well, in the zebrafish community, over 100 mutants. And how we actually do that? Well, the idea is that here we cross this. We would see in a practical course, but we would cross well, male and female wild type fish, and we would inject a mix of Cas9 and single guide RNA, and where we would obtain mosaic F0 larvae, which would have mutation somatically, but some of them will also have mutation in the germline cells. And then arrive to this point where we have adult F0 animals, we would have to screen for potential founders, which are the fish who are carrying the mutation to the following generation. And the advantages of this system is that it's highly efficient, so we really can get up to 100% mutation rate, and it's now, with the proficiency that we got over the years in the zebrafish community, we can really generate, we can obtain like 70% of founder fish after an injection. And the application of this is that when we want to evaluate the loss of functional phenotype for the target validation, genetic target validation, but also we can perform drug screening in these genetic models. And finally, well, the idea is to model human disease, right? And this is the whole pipeline. Well, we have the founder generation that I have just explained you. When we obtain a founder with out of frame mutation that is carried to next generation, we outcross this with dual type fish, and then we will obtain heterozygous larvae. And this when we cross this heterozygous larvae that we have identified to fin clip, so we do a genotype after genomic extraction, cutting the fin of the fish. Then when we have crossing of these two heterozygous animals by Mendelian ratio, we would obtain mutant animals in the F2. Okay, and then in these mutant animals, we can observe the expected phenotype. So just giving an example of some of the work that we have performed. So in this case, we have Parkinson's disease. You all know about that. This is a progressive disorder of the nervous system. And the familiar cases are up to 10–15%. And the hallmark of this disease, it's a loss of dopaminergic neurons in the brain, and the aggregation of alpha-synuclein. And at the moment, there is no cure available. So we thought, okay, let's generate some zebrafish model for that. So the first thing that we did, okay, we identified the genes which are involved, mainly Parkinson's, in this disease, in the familiar Parkinson's disease. And then we checked whether they are autosomal recessive, because we wanted to pursue like knockout approach. These are, all these proteins, they are involved in pathway that then, like disrupting this protein would lead mainly to neuronal death and impaired dopamine release. So we checked if all these genes had an ortholog in zebrafish, and most of them, they had an ortholog in zebrafish with a high homology, sequence homology, except one of them, which is the alpha-synuclein. So we generate knockout for all of them, but for the alpha-synuclein, which had not an ortholog, we generate transgenic line, which is overexpressing the human alpha-synuclein. And then we ran this mutant through a behavior assay. So basically, we use alternated dark and light flashes to analyze locomotor behavior and anxiety. Also, we looked at habituation, doing a tapping on the fish well, and then we tried also to induce possible convulsions with the light flashes. Okay, so this is just to give you an idea of the results that we can get. So here you have like well, different parameters which are analyzed. These, quite few data from quite few mutants. But what you can see here is like the stereotypical pattern of one of the mutants, which is actually disrupted. So we look at the maximum speed, the maximum distance moved, and also in the tapping we look at maximum speed. So here we have like an overview of the all different mutants. Just as an idea, you can say that some of these mutants, they have a behavioral phenotype which is affected, which is somehow recapitulating the behavioral alteration that we can observe in humans. And we also looked at dopaminergic neuron population doing tyrosine hydroxylase staining. Here you can see like dorsal view of a zebrafish larvae with different clusters of dopaminergic neurons. We looked at all these different neurons in the different mutants, and we could see that in some of the lines we had like a degeneration of these neurons, recapitulating also the human phenotype. This was just to show you that we can actually have a recapitulation of human-like phenotypes in zebrafish mutants. But the thing is that, okay, there are some cases when we more or less know already the kind of mutation or the gene that is affected, and we can knock this out. But in some other cases with omic approaches, actually with multi-omic approaches, we have huge list of potential therapeutic targets that we would like to knock out, and this might become complicated if we want to generate mutants for all of them. And so here it comes like the high throughputness also in the genetic part of the screening. So as I said, in the last years, we achieved such proficiency in generating mutation already at the F0 animal that when we injected the Cas9 and single guide RNA complexes, we could distinguish two kinds of population, let's say. A population which has a high rate of mutation, population which has a low rate of mutation, and then we can assume that the population which has the high rate of mutation, it's already de facto mutants in the F0. And this is just an example which was published in 2016, where this approach was first presented, where we had these crispants, which are the somatic mutants, meaning that already in the F0 generation, we have up to 100% mutation. This was a mutant in the TBX58 gene involved in heart development. And the idea is that we would obtain actually different... Well, there is a bit of noise. Wait, can you still hear me?
Yes.
Yes.
Now.
Okay. So the idea is that we would obtain different fish larvae with different degrees of phenotypes, which might be based then on the severity of phenotypes might be based on the mutation rate. And so as I said, this is very important because now we can start from a list of candidate genes, and we can generate crispants, and then we can do a phenotypic analysis and compound screening on these crispants already. The only thing is that indeed the problem is that as it was mentioned before, we always have to look to genotype-phenotype correlation. If we look at the phenotype, then we have to see indeed if this phenotype is led by a high mutation rate. And this is really time-consuming, and we have to go through long genotyping procedure. So we thought, okay, this paper that we actually published this year, we thought, okay, if we have a reporter gene which is that correlates, like whose loss of function correlates with the main gene that we want to knock out, we might really speed up the genotype-phenotype correlation. We might not even need to do that. And so what we did we use a reporter gene, which is a tyrosinase, which is involved in pigment generation. So we assumed that if we knock out the tyrosinase gene and we have unpigmented fish, and at the same time we use another single guide RNA targeting another gene, which is independent, from the tyrosinase pathway, we can actually select a fish who already have high mutation rate, because the unpigmented fish would have a high mutation rate also in the unrelated gene, which is our gene of interest. And first of all, we generated the tyrosinase crispants. Okay, and we checked whether the tyrosinase crispants, this transparent fish, let's say, which have a lot of function in tyrosinase, they would not be affected morphometrically, and this was not the case. So the tyrosinase crispants were normal, and since we were going to knock out epilepsy genes, we also looked at the behavior of the tyrosinase crispants. So we looked at different sensory motor behavior, habituation, and also combustion pattern in the scheme that I've shown you before. And we also checked that tyrosinase crispants alone did not lead to a CNS developmental defects. And so we basically choose a set of genes involved in childhood epilepsy, and then we targeted all of them with this reported gene approach, the tyrosinase gene approach, assuming that the simultaneous inactivation of the tyrosinase gene and the second gene of interest would allow for phenotypic selection, which is based on the degree of pigmentation. So here we have the mutation rate of the non-pigmented and pigmented animal, and then what we could see that in most all of the cases, the unpigmented animal, the tyrosinase knockout gene, actually carried the highest mutation rate in the second gene, which was our gene of interest. This was not the case for SCN1A, because in both cases, the single guide worked so well that, either pigmented or not pigmented, we would have almost 100% mutation rate in this gene. And so we use this in order to then phenotype all these mutants. So we run them through the Noldus system. It's the Zebrafish system to tracking of individual larvae. And we looked to epileptic-like behavior phenotypes, which are mostly maximum velocity and angle turns, if you can think. But the more speed and more angle turns the fish larva has, this can be more or less related to epileptic-like behavioral phenotype, because in order to actually look at seizures, we would have to do like electrophysiological analysis, which we are not doing here. So we are just looking at proxy of epileptic phenotype, and we can see that for most of the genes that we actually test, which are genes relevant for childhood epilepsy, we could see that there was affection in this kind of behavior. And what is the advantage of this? That compared to generation of mutants, indeed, this we can do things in much shorter time. So we can have crispants generation plus phenotyping in 2-3 months. And so this allows us to look at a high number of genes at the same time. And then once we prioritize these genes, we can indeed generate a mutant line if we want to do a more extensive phenotypical analysis. And but we wanted to go like one step further. I said, okay, now the drug screening is performed with the mutant animal, but when we said, okay, but can we use already this crispant in F0 to do drug screening? Because this would even reduce the time of the drug development. And so this is how it usually works. Well, this is for the epilepsy genes. As I said, okay, we do epilepsy gene discovery in patient. We model this in the fish. We do behavioral and electrophysiological assays if you have this available in-house. And then we go through high-throughput phenotypic bus drug screen, finding finally anti-epileptic compounds. And finally, we would bring this to the clinical phase as well. And the idea here is that we use actually one of the mutants that I've shown you, which is a sodium channel. In this case, it has the sodium channel encoding gene, which has been disrupted, SCN1LAB crispants. And we treated this with MSO and fenfluramine at different concentration. And what we can see that while the maximum velocity and angle turns, which are the phenotypes that I told you that are more related to epileptic-like phenotypes, they are impaired in these crispants, so these somatic mutants, we can see that indeed there is a reduction, there is a kind of rescuing of the phenotype with the fenfluramine treatment. And this is just an example that I'm showing you, but we can potentially treat with a huge battery of compounds our crispants of this gene or of different gene. And now, I mean, just in this last part, I've just shown you knockout approaches, which are double-strand break based approaches. And but one of the drawbacks of this is that actually when we do knockout approaches, I mean, we have loss of function, but in some cases we might have some mutation which are gain of function. So we want really to mimic the precise mutation that we find in human into the zebrafish. And then by doing that, using this to do kind of personalized treatment. So, you know, we would move towards personalized medicine. And so these are just few examples of tools that have been developed in the last years. So one is the base editing. So the base editing, you know, we have dead Cas9, so it does not produce double-strand break, and this fused to cytidine deaminase. So we have the deamination of the C. So we can modify a C and convert to other bases, and then we would have basically mutant that is precise mutant on this precise base. This is just an example where basically we have a mutation in the CBL gene, which is involved in the Noonan syndrome, which induces growth defects. And by precisely mutating this base pair, we could see, well, I mean, this is from Filippo Del Bene's lab in Paris, that has been pioneering genome editing approaches in zebrafish. Basically, we can see the same growth defects that we observe in patients. And, you know, virtually, we could use this kind of approach to induce different mutation at different loci, mimicking causative, mimicking human disease. And lately, there is the prime editing approach, because I mean, one of the drawbacks of the base editing is that we can just do some kind of conversion, but with the prime editing, which is still different, we have like a Cas9 nickase, so it just induces a nick, a non-double-strand break, and a reverse transcriptase domain. So in this case, with this PEG RNA, which is kind of modified version of the single guide RNA, we have a kind of template that we provide to a reverse transcriptase in order to repair the DNA, and we can have like precise modification without double-strand break in the genome in the genome location that we want. So if you think about that, this is just an example how we do this in zebrafish. I mean, basically as any other application, we do inject the prime editor, the single, and the PEG RNA into one cell stage, and then we look at the mutation. And this is a really powerful method on which we are working because it can really induce precise DNA edits at any genomic location and virtually of any length. Well, at the moment we are at 5 nucleotide length, but we can go further than this. And this last slide that, we are really moving towards high throughput personalized medicine. These are all different. We are not talking just about a neurological disease, but also cardiac and cancer. So these are just different intervention treatments that are based on zebrafish based screening that are in clinical phases. And so just closing, as I've shown, we have a human disease now. We can do target identification and validation through high throughput crispants generation. We can have genetically modified zebrafish. We can phenotypically validate with high throughput platform. and then we can screen a molecule library, and we can do like a hit confirmation and do a hit to lead. So we have a lead that is the therapeutic candidate that we have, and we can also test this in the same Zebrafish model for toxicity and moving to the other preclinical phase. So I just want to thank my team, especially Flavia De Santis, Irina, and Jessica, who have been responsible of the work that I've been showing. And so I mean, feel free to contact me if you have any question, and I'm happy to take any questions.
Well, thank you very much, Vincenzo. It was very impressive at the talk. I mean, we saw different things like a mix of genetics, screening, phenotyping, all this wonderful stuff. And I'm happy that you talked about epilepsy. So I have some a patient point of view question as an epileptic myself. So as a zebrafish, so I saw that you can screen some drugs, you can screen some, so you also check, I mean, behavior and maybe toxicity. So do you think that we, actually, I have another question before that. Do zebrafish sleep?
That's a good question. Actually, yes. I mean, they do have like that. I mean, they have like circadian rhythm. So we have to keep this rhythm, especially when we do behavioral phenotyping, not just in the adult phase. We do have like 14 hours light and 10 hours dark phases. So I mean, they have quiescent phases, let's say. I mean, we can do, yeah. I mean, we could, yes.
They do sleep, but they don't shut an eye.
Okay.
Yes.
Okay. So then, if they sleep, I do assume that we can perturb the sleeping.
Yes,
And
we could.
Screening stuff. So you think that we can, in the same time, like deal with genetics, with genes, perturb the sleeping, and add the drug that you found. And moreover, we can reach in an age-related way the behavior of the cell. As I told you, it's like a patient point of view question. So do you think that we can, at the same time, perturb not the behavior, but the way of life? Not perturb, but modulate the way of life, the drug, at the same time than the genetic, and see it in a long way term as zebrafish allow us in a short life in a short life cycle. I mean, allow us to see all this.
Yes.
Yeah.
I mean, actually, we can do all of this together as through, the technology that, I've shown you. We can perform this F0 screening, so we can have the mutation, we can treat them with the compound. At the same time, we can perturb with no problem, the light-dark phases, like the circadian rhythm. There are some studies who have been performed which have disrupted like the sleeping pattern of the zebrafish, and they have then looked at inflammation, especially gut inflammation. I mean, you can also look at neuroinflammation. But yeah, this can certainly be done. I mean, there's no problem for that.
Okay. So then now you can add extra information.
Yes. No, as I said, I mean, we have been working actually with Thomas. We started working with the CRISPR-Cas9 in zebrafish like more than 10 years ago now. And the idea is that the CRISPR-Cas9 techniques have been evolving so much that we can really reach a stage where we can have, all together, we can have, already everything in the F0, and we can look how this developed, because they would be, let's say, mutant-like.
Good. Yes, Tom.
Yeah, very nice, Vincenzo. So I have a question about going in a similar direction as the previous one, just for longitudinal studies. So if you pass this point of 5 days, you mentioned before that zebrafish is not, doesn't count as an animal before it starts feeding. So how is it from an ethical point of view and the kind of regulatory mechanisms that are in place if you go beyond this 5-day threshold? And then maybe a second follow-up question as well. So you showed very nicely this locomotor activity as a readout for, like brain disease and neurodegeneration. I wonder how you see the suitability of zebrafish for more complex phenotypes in types of behaviors, maybe also social behaviors and so on. Maybe you can comment on that as well. That would be interesting. your thoughts.
Okay, thank you, Tom. So, for answering to the first question, you actually, starting from 5 days post fertilization, you have to ask for permission to the local regulatory agency. So basically, it's starting from 5 days post fertilization. It is treated as mouse, let's say, so where you have to explain precisely the degree of suffering that the fish, the degree of severity of the procedure, the number of animals that you would need for the entire procedure. So it has to be very well documented. Then it has to go to the internal review of your institute, and then it has to go to the local ethical authority. And then, this takes usually like 3 months from submission. Okay. And then answering to the second question, what I have not shown here, but what we have done, what we can extrapolate from this Vision machine, it's different behavior. So we have like 9 different endpoints. So what we have done, it's like kind of multi-parametric analysis, looking at all these endpoints, and then what we are trying to do now is giving a score to each of these endpoints and doing like principal component analysis to see, okay, this is epileptic, this is actually not epileptic, and then we could use this for a kind of classifier, like machine learning classifier, to say, okay, this is epileptic or no, this is a kind of threshold, and it takes into account a lot of different parameters. As for social behavior in larvae, it can be seen, but it starts more around 3 weeks post fertilization. So we are now developing some high resolution camera tracking in order to track juveniles. And but there are some kind of TMAs that say that you can perform where you can look at interaction between fish. So the repertoire of behavioral assays, it's very vast, and so we are trying to expand this, but also to see how we can look at this from an analysis point of view, in a high throughput manner. Thank you.
Excellent. Thank you.
Hussein?
Yes.
Well, let's start with Hussein and then we move to Marco. Please, Hussein, go ahead.
Yes, thank you. Do you hear me?
Yes.
Yeah, thank you so much, Vincenzo, for this insightful, really good, very good presentation. And actually, I think I'm convinced about the zebrafish as a model. However, I have some question about when you compare to other models, and when you use it for the modeling disease, or as you said, for the phenotype-genotype correlation or drug discovery, I'm asking about the efficiency of translation to humans. I mean, to which level the zebrafish mimic the human physiology, metabolism. I mean, when you have a good result, what is the result that we are perhaps I mean, if the translation is good in clinical training, or when you compare to humans?
Yeah, thanks for the question. Well, this is a question that we get a lot. So basically, when I started working with zebrafish, it was mainly used for developmental studies, but now for also the pharma industry and also biotech companies, they are getting convinced about the translatability of the model, and it is indeed true that the targets, they are very much conserved, and 84% of disease-associated genes, they are very highly conserved. The physiology, most organs, is very highly conserved, and also the development of them, and also the metabolic the metabolism is really highly conserved. Actually, cytochrome is very much conserved, and the pharmacokinetics is also very much conserved. So there is really a high translatability of the results that we get in fish to the human. Actually, in the example that I've shown you, there is mutant of the SCN1LAB, which is the epilepsy gene, where the drugs that have been tested, they did not even go through mice epilepsy model. They went directly to human because this was like repurposing. Most of the drugs that are used are repurposed, meaning that they are usually used for another indication, but now we use them for other kind of treatments. So there is really a high translatability, but we are working in order to increase this and to see actually how this can compare. But the one thing that I want to mention here is that we don't have to get obsessed if we don't find in zebrafish exactly the same phenotype that we find in human, because in the end, we're talking about fish. What is important is that we find something like, meaning if we don't have epilepsy, but we see that the behavior is disrupted, this is already good enough, because this allows us to treat with compounds and see if this disruption of behavior, it is reverted. So I guess that this is really important to understand that zebrafish, it is a proxy of what happens in human. It will not be the same, that precisely the same that what happened in human. So I hope this answered to your question. And we
Yes,
can go.
thank you very much. Thank you.
Marco?
Mohammed, yes, I have seen the zebrafish sleep. Okay, I will only comment to the friend de Donato. And speaking Spanish, sorry. Donato, realmente viendo y escuchando tu conferencia, tu extraordinaria conferencia, bueno, me acerco más a la ilusión de poder reproducir todo ese conocimiento tuyo aquí en Caracas. Un conocimiento que de alguna manera está alineado a tu principio, solo que acá lo estamos desarrollando de manera artificial, lo cual se nos hace bastante cuesta arriba. Una vez más te agradezco tu invitación a compartir esta extraordinaria conferencia y bueno, agradecido. Y un abrazo y espero algún día encontrarnos aquí en Caracas.
Muchísimas gracias, Marco. Thanks a lot.